diversity. What does it mean to be an animal? How do we describe them? What are some key characteristics? And then we have 2 chapters for animals. We have invertebrates and vertebrates. So we'll spend the next 2 weeks at least. Actually, we'll spend the next 2 weeks, next week and this week on invertebrates, and then we'll spend 2 weeks on vertebrates. And so we have a bit of time to talk about animals before we move on to plants. So we are going to start with features in the animal kingdom and things used to classify animals, and then we'll just look at some animal phylogeny a little bit in really broad strokes. We are going to be working at really broad levels when we talk about these organisms. We're not going to be talking about families and genuses of organisms. We're going to be talking about phylums of organisms. So really general, broad categories. Okay, so let's start with what is an animal. Okay, so animals are eukaryotes, right? They have eukaryotic cells. They are all multicellular. So when we talked about protists, we were talking about our single-celled eukaryotes, and some of them were very animal-like. They were hunters and feeders, right? But they were single-celled. All of our animals are multicellular, okay? They don't have cell walls. So plants and fungus both have cell walls. Animals do not. Their cells are simply surrounded by a membrane. And all animals are heterotrophic and conduct ingestion. And we'll talk a little bit about ways in which spiders are kind of an example, or don't directly ingest, but they do still do ingestion. So heterotrophic ingesters means that they have to get their food from other sources, and they have to bring that food into their body in order to digest it. So unlike fungus, which exude their enzymes onto the surface of things and then absorb those molecules in their feeding, all of our animals have some sort of mouth and some sort of digestive tract that is going to be used for breaking down food and digesting it, okay? All of our animals are also mobile. So they're motile, they're able to move. They're able to move, and we're going to see plenty of examples today of animals that don't move as adults, but they were all able to move in their larval or juvenile state. Okay, so some of them sort of settle down and attach to a substrate as an adults and become sessile, but they were mobile at some point in their life early on in their development. Okay. And then lastly, all animals have the ability to respond rapidly to external stimuli. And so they have complex tissues, and some of those tissues serve to respond to stimuli. Okay, when we look at our animal phylogeny, there's sort of a branching point low at the base of our tree of animal life, and one direction goes off into the Parazoan, and the other direction goes off to the Eumetazoan. And the parazoans are really just the sponges. And the word parazoa means beside animal. So zoa is animal. Zoology is the study of animal life. Okay. So parazoans are the beside animal group. They are separate from the true animals because they don't have true tissues. Their level of organization is what we call cellular. And so we'll look at some sponges in their whole, and when we get into invertebrates, we'll actually talk about this. But this is the body of a sponge, and each one of these little structures is a cell. And so they are a collection of integrated cells that function as a single individual, but their cells don't form tissues. So they don't actually have connected tissues. Their cells are independent, and the cells recognize their neighbors through cell-cell recognition. And if you take a sponge and you shred it on something like a cheese grater into a solution of marine medium with nutrients in it, when the cells land into that solution, they'll find their neighbors and reassemble the sponge in the exact same shape it was originally in. So because they don't have like integrated tissues, they still function as individuals, and they recognize each other through cell-cell recognition, so they can actually reassemble and put their bodies back together where their neighbors are. It's not a very nice experiment, but it was done. Okay. All of the other animals are the eumetazoans, and that just means true animals. And so the true animals have complex tissues. So with the exception of sponges, which are kind of out on their own, all of the rest of the animals have complex tissues that participate especially in their coordination activities through the presence of nerve nets or nervous systems, and in their mobility through muscles of some sort. Okay, so these guys, the rest of the animals, have these complex specialized tissues that do specific tasks, like communicate collectively throughout the organism's body or help the organism to move. Okay, and we'll look at those tissues. Most of our animals, as we get above about the back um, 2/3 of that side table, as soon as we move beyond that, they actually go beyond having true tissues and they have complicated organ systems. So really early on, even in our simple invertebrates, we have pretty complicated organ systems. Okay, so this is showing you an image of a snail. And this snail inside its body, here's the snail's mouth. Its digestive system is shown in pink, and its anus is over here. Its respiratory system is shown as in red. Its reproductive system is white, and the coelom is blue here. The nervous system is yellow. And so they actually have distinct organs and complex organ systems, even though, you know, their body is a little bit simpler than the human body. They have a lot of analogous structures, right? They do have some problems, and we'll see this when we look at snails, that their excrement is directly poised to land on their lungs, which is a design flaw in the snail body for sure. The next thing that we want to talk about with animals is reproduction. And for the most part, animals are sexual organisms. They use sexual reproduction as their primary mode of reproduction. Many of them can also do asexual reproduction. So we're going to talk a little bit about both and what are the advantages or disadvantages to asexual reproduction versus sexual reproduction. So in asexual reproduction, big advantage, only one parent. You don't need to find a mate, right? You don't have to find a mate, go through a complex mating ritual, find a mate at the right time. You can just reproduce any old time the conditions are good, right? You don't have to carry around or evolve specialized reproduction organs. You don't have to create gametes. And so you're sort of unburdened of those things. The negatives are, though, that when you carry out asexual reproduction, all of the offspring are clones of the parent. So they have the exact same traits of the parent. There's no chance for genetic recombination. You don't get to roll the dice and see how the genes land in the next generation. You just make a clone of the parent itself. Okay. You can have lots of offspring in a short time. That's a good thing. Generally, asexual reproduction is favored in stable or predictable environments where there's not a lot of change. Conditions are always pretty much the same, and organisms can kind of just depend on the conditions being the same. Anytime you have instability or you need to respond to environmental changes, you want to have that potential for new genetic material to arise, and new gene combinations to find themselves. And so sexual reproduction is favored in those scenarios. And as a general rule, evolutionarily, sexual reproduction is favored. It's evolved independently in many phylogenetic lines, repeatedly over time. Generally, you have more opportunity to survive in your environment through sexual reproduction than asexual. But let's look at some of the ways asexual reproduction happens. I did not look at all of our hydra today, so we may have some that are actually carrying out asexual reproduction. If you find one, let me know and we'll share it with the whole class. Hydra reproduce asexually by a mechanism known as budding. And budding is when you take the individual's body and divide it into more than one body. And this division is usually unequal. And so when a hydra goes through budding, it starts by producing a little bud on the side of its body. And this is just a small clump of tissue. And then it's going to grow and develop that clump of tissue into a larger and even larger clump of tissue. But ultimately, the new hydra that drops away from the parent is much smaller than the parent. So the parent has taken some of its resources and put it into building this small mini-me clone. And then mini-me drops off and goes off and lives its own life. Okay. This is, again, you're making a complete clone. So you're just making an individual that's exactly like you, has your exact same genetic material. You've got some extra resources, so you're going to make a new offspring that has your same genetic identity. So starfish are also able to reproduce asexually, as are planaria that we're going to see today. And I think at least one of our planaria might be in the process of dividing. He looked funny under my naked eye. So fragmentation is where the body is broken and regenerates the missing parts. Okay, so in the case of a starfish, since Tess was asking about starfish, starfish can regrow all of their arms, and they can regrow an entire starfish from a single arm. So if you have a starfish and you cut off one of its arms, it will grow a new arm, and the arm will grow a new body. They can just do that. They have the regenerative ability. So they can go from one individual to two individuals just like that. My favorite story about starfish is that fishermen were getting upset by starfish eating too many mussels and damaging the harvest. And so they started, whenever they caught a starfish in their net, they would chop it up into pieces and throw it back in the ocean. Which just feels like the best karmic comeuppance, right? So instead of eliminating their starfish, all they did was rapidly exploded their population, which is like what happens when you do stuff like that to nature, or what should happen. So they can be damaged. They can't come back from like a starfish can't come back from every damage. You actually have to have enough of the central ring canal intact for regeneration to happen. So it's not like you can't destroy them. There are some invasive starfish that are sometimes referred to as crown of thorns that are invading like the Hawaiian Islands. If you go snorkeling in the Hawaiian Islands, the dive master might give you a knife and ask you to chop up starfish, but you actually have to like fully destroy them and kill them. You can't just chop them up because you'll propagate them if you're not careful. Yeah. They destroy the reef. They're an invasive species and they eat a bunch of things and completely mess up the ecosystem. Yeah. So the planaria we're going to look at today also are able to regenerate. If you cut off the head, the middle, and the tail, each one of them will generate a new piece. They can also split down the center, so you can like nick them between the eyes, and they'll just split into two pieces. And they can do that. They can make the choice to do that. They can split themselves. They usually divide down the center, but they can also reproduce sexually, as can starfish. So hydras can reproduce sexually. Starfish and planaria can reproduce sexually. Even though they have this asexual mechanism, they also can reproduce sexually. Okay. I don't know how planarias experience pain. They have a nervous system, so but they also do it themselves. And so if you're like the hydra, if you're budding, I think that you would probably, create an abscission layer and drop off. But I don't know exactly how they do that. I'm sure that they can. Yeah. Whether they choose to or not is the question. Yeah. And we'll see plenty when we're working our way towards it, but plenty of invertebrates can actually sexually reproduce with themselves. So yeah, that's just like your clones reproducing, only you're saving out an extra step. Right? So another type of asexual reproduction we see is parthenogenesis. The word parthenogenesis, genesis means beginning. So this is a virgin beginning. We don't need to provide our human perspective to that. Basically, what this means is that unfertilized eggs can produce a complete functional individual, and that individual is almost always male. I don't know of any cases where it's not male. And so an unfertilized egg will become a male, a fertilized egg will become a female. And there are some species that will sort of do this parthenogenesis as the rule. And then when environmental conditions become bad or resources become scarce, they'll shift into sexual reproduction. This occurs in a number of different invertebrates, but it also interestingly occurs in reptiles, amphibians, and fish. And so it also occurs in some vertebrates like this whiptail, which is shown here. You sometimes hear about this in zoological collections. You'll hear that the snake at the zoo gave birth and the snake's never seen a mate of its own kind. Well, the snake's been sitting around for 20 years hoping to get its reproduction done, and it hasn't happened, so it goes through parthenogenesis. That when we've seen it in some of those species, it's been like in an extreme case like that where this individual has never been able to find a mate, and so they're trying to figure out a way to propagate. Okay, And then that brings us to sexual reproduction. So we're going to talk a little bit about sexual reproduction generally. And then I'll just kind of like sprinkle in fun facts about organisms as we go through in terms of whether they reproduce sexually or asexually. I'm going to use the terms monoecious and dioecious. Mono means one, di means two. So monoecious means in one house, and dioecious means in separate houses or in two houses. Okay, so what that means is an organism who is monoecious has all of its sexual reproduction equipment in one house. So it's a hermaphrodite. Dioecious means that you have separate houses. So you have male individuals or female individuals, or individuals that do both or switch back and forth between those two and are able to come together separately. Okay, so we'll use those terms to refer to whether or not you have all of the possible reproductive structures in one location or whether you have them in two locations, two separate individuals. Okay, we talked a little bit about the continuum of sex when we were talking about sexual selection, and we'll mostly just default to a binary system of talking about males and females, but that is a very generic layover, like over top of all of animals, because really many of our individuals are males at some point in their life and females at another point. They might be both. There are lots of exceptions to the rules. So when I'm talking about males and females, I'm really talking about individuals who have tissues that produce eggs and individuals who have tissues who produce sperm in the animal world. So we have sperm tissue and we have egg tissue, and we bring those together in some assemblage of bodies. Okay. In order to do sexual reproduction, you have to have these specialized tissues. You have to have tissues that create eggs, and you have to have tissues that create sperm, and you have to house them somewhere. You also have to go through meiosis. And so when you do asexual reproduction, you don't carry out meiosis. You usually are just reproducing through extra tissue, so just mitotic divisions, making a clone, right? And we have these two types of gametes, and those gametes are haploid in the animal world. When they come together, they make diploid offspring. Okay. This is going to be really important evolutionarily because it results in new combinations of genes. Every time an individual makes egg and sperm, they're only putting part of their genes into those gametes. And so when those gametes combine with another gamete, then they're reshuffling the genetic information. So even if you're reproducing with yourself, if you're sexually reproducing with yourself, you've made an egg and a sperm and you've put different contents into those two gametes. And when you bring them back together, they have a new combination that is different than the parent, right? So you're avoiding cloning by recombining the genetic information. Asexual reproduction produces a clone. Like statistically, you could produce a clone through sexual reproduction with yourself, but the chances like 0.00000, like all the way out to almost never. But yeah, theoretically, it's possible. Mathematically, no such thing. Well, your little brother is another recombination of your parents' genetics, right? It's not necessarily the same as you. So siblings each get 50% of their genetic material from each of their parents, right? So you got half of one of your parents' genes and half of your other parents' genes. The set that you got may have a lot of overlap with your sibling, but may have very little overlap with your sibling too, just depending on all the possible combinations of those genes. Okay, so when we use the term monoecious, we are talking about organisms that are true hermaphrodites. And I know that this is a term that a lot of people don't want me to use because of where we're at in our human culture. But the problem with our human culture is that we've applied the term hermaphrodite to humans when that doesn't happen, but it does happen in the animal world. And so the term hermaphrodite is the correct biological term for an organism who produces both eggs and sperm in the same body. Okay. So we have lots of organisms that are indeed hermaphrodites who are capable of producing both eggs and sperm in the same body. And so we can also use the term monoecious, one house, right? They have the tissue necessary to produce eggs and the tissue necessary to produce sperm in the same body, in that single body. So This allows individuals to carry out self-fertilization or to mate with another individual, depending on their reproductive strategies. And sometimes you might reproduce by yourself if you're by yourself, but you might reproduce with another individual if one is available, right? Just depends on the environment. In this picture, we have two earthworms, both of whom are hermaphrodites and who are mating. And both of those individuals are serving as male and female sperm and egg producers in this interaction. So this individual is releasing eggs and sperm, and this individual is releasing eggs and sperm, and they are cross-fertilizing each other, and they will make two batches of eggs from that interaction. Okay. They each are going to have fertilized eggs, so they are each being both male and female in their gamete production in this interaction. And that's standard for earthworms. Like when you see earthworms mating, they're generally both producing egg and sperm simultaneously. And we could go through the whole. I won't go. There are lots of diagrams. If you Google earthworm reproduction, I'll walk you through how that happens. Okay. When we talk about separate sexes or use the term dioecious, two houses, then those specialized reproductive organs are housed in separate sexes. And this can be controlled by a lot of different things. In many organisms, it is controlled by genetics. So mammals, we have an XXXY system where you generally have egg-producing tissue if you are XX, and you have sperm-producing tissue if you are XY. In birds, it's ZZ-ZW, and so sperm-producing individuals are ZZ, and egg-producing individuals are ZW. And then we have these haploid diploidy systems, things like wasps and bees, where all of the sperm-producing individuals are haploid and all of the egg-producing individuals are diploid. And those males come from unfertilized eggs, like our parthenogenesis. This might change depending on the environment. So in many reptiles, actually, whether you are an egg-producing individual or a sperm-producing individual depends on the temperature you're at as you're undergoing your embryonic development. And so temperature can control what kind of tissue develops. And then some individuals change throughout their lifetime. So, and depending on the species you're looking at, some species might start off life as a female and switch to a male. Other individuals might start off as a male and switch to a female, depending on cues in the environment. Some individuals might never switch, and other individuals might switch. And so it totally depends. There's a lot of species-specific examples of alternating. So there is a physiological change, and the example that I have up here of these guys is one where it does change. And so they do have like high levels of sexual dimorphism. And the individual actually completely changes its appearance as it goes through that process. And some of these, it's like there's one male in the population, and so the male dies and one of the females takes over, or vice versa. Kind of depends. This is pretty common in fish. These alternating systems are, I think all of the examples I can think of off the top of my head are fish examples. Turtles are a really common example of the environment. And it's like, we're talking about like, 0.2 degrees Celsius or 0.1 degrees Celsius temperature change in the eggs is enough. So if you think of a turtle, a sea turtle laying their eggs on the beach, the eggs that are closest to the sand are going to be warmer than the eggs are at the bottom of the nest. And so you have one temperature eggs on the upper layer and one temperature eggs on the lower layer or on the outside of the nest versus the inside of the nest. Okay. There is in mammals, which is totally separate from this, there are environmental changes that happen, like with position in the womb, for example. And so animals that have multiple litters, depending on your position, you can experience greater nutrients or less nutrients or things like that. We talked a little bit already about external fertilization, right? We talked about external fertilization with gametes. Yes. Remembering which class I was talking to. When all of the eggs and sperm go out into the water, Yeah. So external fertilization is generally the norm in aquatic environments. And so egg and sperm are released into the water. We were talking about this a little bit with gametic incompatibility with urchins and other reef dwelling organisms who are sessile or don't have high mobility. They just put their gametes out into the water and the gametes find each other in the water. This also happens with many fish where the eggs are laid and the sperm are cover the eggs after the eggs leave the body. You also see this with many amphibians. And so if you look, most mollusks, not all, crustaceans and echinoderms use external fertilization, as do most fish and many amphibians. Right. So this is definitely a place where gametic compatibility is required. Yeah. So if there's incompatibility, then their fertilization won't occur. Yep. Okay. And then we have internal fertilization. So most land-based animals are going to use internal fertilization, although some aquatic animals also use internal fertilization. And internal fertilization does not always require copulation. Did anybody listen to the Radiolab Beauty Puzzle yet? One person. Like why birds lost their penises? It's pretty cool. You should listen to it. If I hadn't enticed you with the beauty puzzle, it tells you why birds don't have penises. Why a lot of birds don't have penises. Copulation is not required for internal fertilization. Most birds, for example, don't have copulatory mechanisms. Many insects also don't have copulatory mechanisms but have internal fertilization. This salamander up here, it's almost spotted salamander season. This is how the spotted salamander does internal fertilization. These are spermatophores, and spermatophores are essentially sperm packets, and the sperm is actually housed in a gelatin packet and secreted external to the male's body, and the female actually picks it up and inserts it herself. And so this is like acquiring a spermatophore. You see this in many insects in particular. The male may actually hand the spermatophore to the female or leave it on a branch or something, and she can pick it up and take it or not, depending on her choice. Oh, so birds who don't have copulatory structures rely on what my professor always called the cloaca kiss. The cloaca kiss. So they have a single opening for excretion and also for reproduction, and they touch that opening called the cloaca together. And so just the touching of the opening is enough for sperm to be transferred. Yeah. So there's no copulatory structure necessary. They just briefly, very briefly, press their cloacas together, and that gets it done. Okay, so let's talk a little bit about what happens when those eggs are fertilized, how those eggs are treated. And so we have some terms for whether the eggs are laid external to the body or the eggs are housed within the body. So oviparity, this word oviparity, means that once the eggs are fertilized, they are laid outside of the body, or in some cases, they were fertilized outside the body already, right? So a bird's nest, classic oviparity, right? The eggs are laid outside of the body and the embryo is nourished by the egg's yolk. So during its embryological development, it's feeding off of the yolk of the egg, and that's its supply of nutrients. That's how it gets all of its needs met. And then when it's reached the end of its embryological development, it hatches and leaves the egg, right? These eggs can be aquatic or terrestrial. So many fish use oviparity. This fish is mouth brooding, and so mouth brooding fish are really cool. The eggs are actually fertilized externally, and then in many cases, the male picks them up and carries them around in the mouth in order to protect them from potential predators. They don't feed during this time. They literally just swim around, like incubating the eggs and keeping them safe until they hatch. But this would still be oviparity because the eggs are external to the body, they're laid external to the body, and they develop external to the body. Okay, they're not being housed inside, even though he's carrying them around in his mouth. So gastric brooding, mouth brooding, and also skin brooding. Some eggs actually, the animals store them in deposits in folds in their skin while they develop. Some frogs do that, yeah. Hopefully not. I'm sure it's happened. Oops, but Well, it would get digested. If it ends up in your digestive system, it's just food. Yeah, you just accidentally ate your offspring, but I mean, it still gets digested. Okay, then there's ovoviparity. So these words are really similar, oviparity and ovoviparity. So ovoviparity is fertilized eggs are retained within the body. Okay. And then the developed offspring hatch and leave the body. So live bearing fish like guppies, this guppy here is full of little baby guppies. The embryos are still nourished by the yolk inside the egg, inside the parent's body. And so they're developing inside their egg. They're feeding off of the yolk. When the yolk nutrients are done, that's the end of the nutrients. They can't get nutrients from the parent's body. But when their development is done, the young actually hatch and leave the parent's body. So this is a guppy giving live birth. So here are little baby guppies swimming out of her body after they've developed inside. So this is actually inside the female's body. Yep. And so the female is fertilized. The eggs remain inside the body and they develop inside the body. Nope, we're this. So viviparity, or being viviparous, is when young develop within the parent, that should say within the parent, receiving nourishment directly from the parent's bloodstream. So mammals, all mammals, well, that's not true, duck-billed platypus and anteater. Most mammals are viviparous, which means that the young directly receive nourishment from the parent's bloodstream. And so here is a human offspring developing, right? And this is the placenta, which is an organ that the mammal grows solely for the purpose of nourishing the offspring. Right, so they grow a whole new organ, increase the circulation of the body by 30% in order to feed this thing, right? So basically, mammals are parasites. They're like feeding directly off of our bloodstream, right? The offspring. You also see this in fish, some fish. This is a shark giving live birth. And this shark actually has a structure to feed the baby sharks. They are attached through an umbilical cord to the parent's body. So we think of that as being a really uniquely mammalian thing, but it's also evolved separately in sharks. If you're a land animal, this prevents dehydration. So you don't have to worry about whether or not your eggs dry out because they are carried inside the aqueous environment of the parent's body and they're kept safe as long as the parent isn't encumbered by carrying the offspring to the point of being super vulnerable to predators. The offspring are kept safe. And in fish and these sharks, it's protecting those embryos from predators and making sure that they get all the nourishment that they need. Yeah, so kangaroos are also considered viviparous. They just have an early end to the embryological development, and the offspring finish nursing attached to a nipple as opposed to attached to the bloodstream. Yeah. Okay. So this is our basic pathway for animal reproduction. We have an egg and a sperm that are produced either by separate individuals or by the same individual, and the egg and sperm meet, right? Fertilization occurs and produces a zygote. The zygote is that single-celled fertilized egg. And so the zygote is then going to begin to divide. And so one cell is going to become 2 and 4 and 8 and 16 and 32. And so we see this cell dividing. And in this diagram, this size change is correct. As that egg begins to divide, it doesn't actually, the mass of cells it forms doesn't get bigger than the original egg. At first, we begin by dividing up the egg itself. And so we're making many small cells. And then at some point, that ball of cells, the cells continue to divide and the cells begin to push to the outer edge of the ball. And so we end up with this hollow ball of cells, and that is known as the blastula. So we start with our zygote. The zygote is a single fertilized cell, and then that zygote begins to divide and divide and divide and divide, and that single cell becomes a ball of much smaller cells that is the same diameter as our original egg. Okay, those cells push to the outer edge and form the blastula, and we have an opening in the center here. That's called the blastocele, and we're going to come back to that term. But now we have a hollow ball of cells with an opening in the center. That opening in the center, as the blastula develops, we start to fold some of those cells into that opening, and that process is known as gastrulation, and this becomes the early gastrula. It's starting to get bigger, and we're starting to fold cells into the interior, and then we're going to fold cells into the interior even more and get a little bit bigger. And this is our basic development that all animals go through. Okay. This gastrulation process and the formation of the gastrula is where we begin to create the digestive cavity. And so this little loop here that gets pushed in is ultimately going to become where digestion occurs. Okay. Then we have our, in the case of our starfish, our young larvae, we would form an embryo in other animals. That larvae, in this case of the starfish, is a free swimming little critter that goes out and lives its life as a larvae swimming around in the water. And then it undergoes metamorphosis to become the adult form. Okay. Not all animals go through metamorphosis, but many of them do. And during metamorphosis, the tissue is rearranged into a new form in order to produce the adult form. Okay. So now we want to talk a little bit about, we're gonna come back to this process. But before we do this, I want to mention that the set of genes that controls this process and controls the development of the organism during its embryology, during its larval form, and ultimately to adulthood, is a group of genes known as the Hox genes. And this is a set of genes that contain the structural genes for animal bodies. And they are remarkably homologous across groups. And so we have really, really similar gene sets in all animals. And these Hox genes essentially work like master switches to turn genes on and turn genes off in order to control development. So ultimately what these genes do is they control how many appendages the organism is going to have, where those appendages are going to be placed, how many body segments the body is going to be divided into. All of those kinds of things come from these underlying structural genes. And the combination of Hox genes that are on or off controls how that body develops. So when we talked about comparative embryology and our evidence for evolution, we said that it's really hard to tell a chick embryo from a pig embryo, from a frog embryo, from a human embryo early on. That's because that development is all controlled by this really, really similar set of genes. And it's just that as those genes are regulated, we start to see movement towards different ultimate body morphs. And so this picture is just showing you Hox genes on a single chromosome in a Drosophila fly, and then on 4 separate chromosomes in a developing human embryo. And the color coding shows you where the gene is the same. And so this pink gene is found on the Drosophila, and on 3 of the human chromosomes is a gene that's analogous to that, that's controlling similar things. And you can also see where those genes act in the development. So anterior to posterior, appendages here has the purple genes, the appendages are green, the green genes, which are part of the spine here. So you can kind of look at that overlap. So this is a place where we have a lot of homology in embryological development across the entirety of all animals. Okay. All right, so let's talk a little bit about how we classify animals, and then we will take our break. So I just want to give you some terms that when you're talking about animals, you can use to describe their bodies. And so the first terms I want to give you are dorsal and ventral. You guys have probably heard these terms before. I'm going to use our dolphin as an example. The dorsal fin is spine side. Okay, so in anything that has a spine, the dorsum is the spine side, and the venter or the ventral side is the belly side. So whether you're an organism that's on all fours, that swims, or that upright walks, you can keep track of the belly side versus the spine side, right? So the dorsal side is the spine side, and the ventral side is the belly side. We don't see these terms with our earlier invertebrates that have spherical bodies, right? Or that like something like a worm. You often don't talk about dorsal and ventral surface, except for when you have clear differentiation inside the body, like where the nervous system is. And so often, like in a worm, the nervous system is actually ventral. It's on the belly side, it's on the ground side of the organism. But things like starfish are mouth up, and so their mouth is on the opposite side you expect it to be on. And so these terms work for most invertebrates, but not for all. And so, but we'll use them when we kind of want to talk about the upper surface versus the lower surface. And then we also will use directional terms for sort of the forward-facing side where sensory organs are, and the trailing side where propulsion or excretory organs are. And so in our dolphin, the head is the anterior and the tail is the posterior. Okay, same is true with yourself. This is the anterior of your body. This is my posterior, right, the bottom half of your body. You can take an organism and move it from horizontal to vertical, or even some of our organisms that are more radially shaped, and you can apply these terms. Okay. Typically, the anterior is associated with cephalization if cephalization occurs. I think I took that slide out. Cephalization is the presence of a head. We're going to start with some invertebrates that don't have a head. And as we move up, we'll see that you have a concentration of ganglia or sensory organs in a region, and that leads to what we would refer to as a head, or the presence of cephalization. Okay, we also are going to talk about organisms in terms of their body symmetry. So is the body radially symmetrical or bilaterally symmetrical? And what that means is that you have matching or similar body parts across an axis. This does not apply to our sponges, or parazoans, because it's not found in animals with tissue level, with subtissue level organization. We only see it if you have true tissues. So the sponges don't have any symmetry. They're considered asymmetrical. But our anemone here has radial symmetry. You can divide its body into matching parts, almost like around the spokes of a wheel. And our frog is bilaterally symmetrical. You have matching sides, right? So you draw an axis down the center of the body right between the eyes. You will have matching parts on either side. So radial symmetry is common anytime we have body parts radiating from the center. So we have an organism that is roughly spherical in shape and it has matching body parts around that sphere. And these tend to be things that are sedentary. And so radial symmetry works really well if you're a sedentary anemone anchored to a reef somewhere and you're kind of hanging out and you can't move, because now you can sense your world from all the way around your body, right? So you're able to receive stimuli equally from all sides. You don't have to worry about whether it's behind you or in front of you because your sensory organs are radially symmetrical. They're around the entire body. Okay. This occurs in organisms that have two germ layers, the ectoderm and the endoderm, and we're going to talk about germ layers in just a second, and so you'll know what those are. But if you have two germ layers, you tend to be radially symmetrical. Bilateral symmetry is where your body parts are arranged in mirror images across the center axis. So you have a right and left side. Okay. This occurs when you have three germ layers, and so you add the mesoderm. We'll talk about that in a minute. Our crayfish here has bilateral symmetry. We can clearly see a dorsal surface and a ventral surface, an anterior end and a posterior end. And we can divide that body right down the center and see the two sides, right? Did anybody see on social media the cardinal that was going around that was half male and half female coloration? That picture that was out and was split kind of right down the center. This symmetry also has to do with how that embryo develops. So early on in its cellular stages, as we start to push the gastrula in, we sort of split off the sides a little bit. And so you have this sort of separation, even that early on in development, where you've got these two sides, bilateral symmetry. Do you have a question? Probably. Yeah. And some of those colorations, especially in things like cats, like a patch of cells will have a set of genes turned on, and so all of the neighboring cells are there. So if that split occurs early, then whatever went here is going to like radiate the patch of cells nearby it with its coloration, whereas whatever is over here is doing its own thing. Like calicos, those patches are actually associated with the original cells during early embryology. What got turned on or turned off in those cells then gave rise to everything around it. So octopus is a great question because it seems kind of radial, but octopi are actually bilateral. They have cephalization, they have eyes, and we split them down the center. Their tentacles actually pair across, even though they seem radial. And generally speaking, our radially symmetrical organisms are pretty early on in the tree of life, except that then we see really close to vertebrates, we see a group of radially symmetrical invertebrates that are further along. But that's their symmetry is actually secondary. They have bilaterally symmetrical larvae and radially symmetrical adults, so like starfish. They're bilateral as juveniles, radial as adults. So I'll just give you a quick example. Beetle is bilateral, a coral is radial, and then a sponge has no symmetry, right? Although sometimes sponges appear radially symmetrical, it's kind of just by chance because they develop in a way that is free of any tissues at all. Okay. Oh, there's cephalization. I just moved that slide. I thought I deleted it. So I will let you spell that word out. Cephalization that I keep talking about just refers to the concentration of sensory organs in a head-like region, right? So we have a very defined head region. When you look at a leech, it's hard to know which is the anterior and which is the posterior. If it attaches to your body, you're like, okay, well, now I know which end the sucker's on, right? But when you look at a leech, if you have it in your hand, you might not be able to tell. But if you were to dissect it, it has a clear ganglia and nervous system concentration in the anterior end. And so although it doesn't have like a visibly obvious head to you, it does have cephalization and has a head region in terms of where its sensory organs are concentrated. Okay. All right, so let's talk about embryonic germ layers in our last little bit of time here. Embryonic germ layers are layers of tissue that give rise to different parts of the body. So my little circle over here that looks like, boy, you step back, it looks like a fisheye. It's not supposed to look like a fisheye. It's supposed to look like a baby embryo. So our little circle here is like that little clump of cells developing into the blastula, right? And what we're looking at are the different layers of that the cells can start to take in that early phase as we move from blastulization to gastrulization and start to build the center body cavity. Okay, so in the center in black is the gut cavity, and the gut cavity is basically an open space. Like if you think about your own digestive tract, you have a series of tubes from mouth to anus, right? It's a hollow tube where the food moves through. The gut cavity is an open space or a hollow space where food can go in for digestion. Okay, so that's open. There isn't tissue development inside of there. That's an open hole. And then in yellow, the layer closest to the gut, we have what's called the endoderm. And so endo, inter, into the inside, that term endo, like enter, is the innermost layer of cells. And it is going to be directly around that gut cavity. So it's going to be the lining of the gut cavity. It's going to surround that hollow organ. And it's ultimately going to give rise to many of our hollow organs, as well as our respiratory surfaces. So it's going to give rise to gills or lungs, depending on the organism, and it's going to give rise to hollow organs. The ectoderm on the other end over here in red, so the outermost layer, the ectoderm is on the outside. That's the outer layer. It's ultimately going to give rise to the epidermis, the endodermis, the outer layer, the covering of the body. It's also going to give rise to nervous tissue. So brain and nerves and spine will all come from ectoderm germ layer. So if you're radially symmetrical and you only have two germ layers, you have endoderm and ectoderm. If you have a third layer, like our bilaterally symmetrical organisms do, that third layer is mesoderm, meso in the middle. Okay. Helps you move. Yep. So the mesoderm is shown in orange, and this layer is the layer between the endoderm and the ectoderm. It's the middle layer of cells, and it gives rise to muscles, to the circulatory system, and to the skeletal system. So this is our mobility, some of our connectivity. It also is going to provide anchoring, and so it's going to help anchor organs in positions relative to each other. It provides a lot of our connective tissue. So it's going to be responsible for mesenteries, which help hold your organs together. And so this is sort of the middle layer that holds the things together in the middle. Um, Always a possibility. I might have to pick one that's better than my weird PowerPoint created fisheye. I don't think I've ever stood this far back from it, and I'm a little bit surprised how much like a fisheye it looks. I might be more likely to use one of these stock pictures. Okay, so if we're looking at the development of our little blastula, our lump of cells, so this circle here, our blastula, is made up of many, 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 many cells, right? Our embryo's been dividing, we've got lots and lots of cells. The cells have pushed to the outside edge, and now we have a hollow ball, right? So at this point in human development, the hollow ball is still floating around. It hasn't even embedded itself yet into the uterus. It's still bopping around in the fallopian tubes or cruising around the uterus looking for a good spot to settle down, okay? All the cells are pushed out to the edge. We've got this opening. And then our blastula, as it starts to move towards the gastrula stage, a section of those cells in the blastula are going to begin to become the endoderm. And they're going to push in and begin to line our little blastula. That process is going to now create two separate layers. We have the outer layer, which is our ectoderm, and then we have the inner layer that's starting to push into the center of the ball of cells, right? And as that begins to push into the center of the ball of cells, it's going to ultimately completely line our little blastula, which is now going to be called a gastrula because it now has the gastric cavity. Okay. So in this organism, we have our gut cavity or gastrocele. Cele means opening or space. So we have our digestive space. We have our endoderm surrounding that space. We have our ectoderm surrounding the outside of the animal's body. And then we have an opening. That opening was created by the folding in of this endoderm. It's called the blastopore. And the space between these two layers is ultimately going to become what we call a body cavity, a space in the body, right? All animals start this way. You too were once a little blastula, right? No, this is many cells. This is tissue. This is the ectoderm tissue in orange and the endoderm tissue in red. We're going to get there. Give me a minute. We're getting to 3 germ layers. Now I'm going to go ahead and go a couple minutes over and finish this thought, and then I'll give you your break. So those organisms that only have 2 germ layers are diploblasts. So di for 2, blast like the blastula. So diploblast, 2 blasts. They've got 2 layers of tissue. And these tend to be radially symmetrical organisms. And what you're going to see when we start to look at those radially symmetrical organisms in more detail is we've got our gut cavity here. This is, should say gut cavity really, in the center. Our endoderm is now in blue. Our ectoderm is in yellow. And then there's this non-living fluid-filled space known as the mesoglia. And if you think of a jellyfish swimming, the bell of the jellyfish is all mesoglia. It's just like this gelatinous stuff that the body can push its muscles against. Okay, so this, in a radially symmetrical organism, there's this non-living ring between the endoderm and the ectoderm. So that body cavity gets filled in with non-living stuff, usually a jelly-like substance. It's a divider. It ends up being, it creates a hydrostatic skeleton. It creates a way to move against the body. It provides some support and structure to the body. The mesoglia, to some extent. Yeah, in some cases it's quite large, like in a jellyfish, it's really large. In other cases, it's like a very thin, non-living layer between the two tissues. But it's where those tissues grew into this space, and there's a little gap left. And that gap gets filled by a non-living space known as the mesoglia, which is usually fluid or gelatinous filled. Okay, then we have triploblasts, and these are animals with 3 germ layers. So these guys have bilateral symmetry. They have the endoderm and the ectoderm, and then that space, that original space, gets filled in by mesoderm during embryological development. So instead of having a non-living space there, that space gets filled in with the 3rd germ layer, the 3rd tissue type, the mesoderm. Okay. Do you have a question? Yeah. We are this one over here. So a long time ago in your early part of your baby book, you looked like this. You had a little gut cavity in the center of your gastrula. You had an endoderm, a mesoderm, and an ectoderm. And then that lump of cells starts to take on a shape. And I always think that the early embryo looks a little bit fish-like because it's got that like rough edge. But then it starts to go from the ball of cells to cells that have to a structure that has more defined shape. But we really all are just little balls to begin with. So the ball is going to grow by creating more cells, but then it's also going to start to change shape. And so obviously we are not balls as adults, right? And so early on in the embryological development, some of these cells out here on the ectoderm start to form skin and brain and spinal cord and nerves. And some of these cells in here break off and start to form hollow organs like hearts and lungs. And okay, so when we are going through this development, I'm going to just do these last couple slides and I'll stop. Another way that we classify animals is by whether or not they have what we call a coelom. And so those animals that have a true coelom are called coelomate. And coelom is a word for opening. So remember I said that something had a gastrocele or a blastocele and it was just an opening or a space. Coelomate means that they have a true space in the body cavity. And that space is completely lined by mesoderm tissue. So what happens is as this organism is developing, and this is a earthworm, now we keep changing colors because I'm using all these different diagrams. The digestive tract that is here in yellow. This was our original endoderm tissue. Our body coating, our ectoderm, is out here in blue. And then the red is the mesoderm. And as the mesoderm grew, it divided so that it completely lines the ectoderm and completely lines the endoderm. And there's some space between those two linings. Okay. So most of our upper organisms, like up in the top of the family tree, are coelomate. The mesoderm completely surrounds the gut cavity. It completely lines the ectoderm, and then there's a space. And that space might be fluid filled. It might be a space where organs are anchored and held in place, but there's a space in the body cavity. That is us. We are coelomate. We have multiple spaces. We have the space around the lungs and the chest. And then we have the diaphragm that divides. And then we have our second space down here, and we have a space around our brain cavity. So we have multiple body cavities. And tissue and yeah, stuff. Not a lot of fluid. I mean, it's not like we don't pour out fluid like some animals, but we have interstitial fluid surrounding all of our tissues and organs. Yeah. The next one down, I'm doing these in opposite of evolutionary order because I think it helps to define what a coelom is first, is something that is pseudocoelomate. So pseudo meaning fake or false. So this is a false coelomate. This animal has a false coelom, which means it's partially lined by mesoderm tissue. So here's our endoderm, here's our ectoderm, and here's our mesoderm. And the mesoderm lines the ectoderm, but it doesn't surround the gut cavity. So what does mesoderm become again? Does anybody remember? Muscle, right? So if you're coelomate, you can squeeze stuff through your digestive tract using your muscularization. If you are acoelomate, the only way to get stuff to move through is to put more stuff in, food out, right? You don't have muscularization of the gut cavity. So there's a disadvantage there. By not having a complete coelom, you don't have the muscularization of the internal gut cavity. You only have the muscularization that allows you to move your body. So this is a roundworm. Roundworms have this. Roundworms are the classic example because you can just slice through their body and look. It's nice to be able to compare the three worms. The segmented worm, like the earthworm here, is fully coelomate. The roundworm, like the nematode, is pseudocoelomate. And then my favorite worm that we're going to look at today because they're so cute, the planaria, are acoelomate. They don't have a coelom at all. There's no space. Instead, the mesoderm completely fills the entire space between the endoderm and the ectoderm. So there is no body cavity. They just have mesoderm tissue filling that space. So both of those things are next steps evolutionarily. Like there's having a complete coelom and there's being segmented. So our bodies are segmented. A grasshopper's body is segmented. It's segmented in a different way. We don't have individual rings like an earthworm, but segmenting and redundancy are actually advantageous. And you see those in organisms that are further along or more derived on the family tree. And you see the lack of that further down. And so they correlate in that they're both big steps evolutionary that happen simultaneously. Coelomate possess a true coelom. The coelom is completely lined by mesoderm. So mesoderm around the endoderm, mesoderm around the ectoderm. Pseudocoelomate has a false coelom. There is a space, an open space in the body cavity, but the digestive tract or the endoderm is not completely lined by mesoderm. And then, do you have that, Josh? Acoelomate does not possess a coelom at all. There is no body cavity. The entirety of the space between the endoderm and ectoderm is filled with mesoderm. Okay. One of the functions of the coelom is that it can function as a hydrostatic skeleton. So if that space is filled with a non-compressible fluid, you can contract your muscles against it. And this allows organisms to move. So you're going to explore that with our hydrostatic skeletons in lab today. Man, that one's done too. I bought these fancy skeletons and they break all the time. So we've also got water balloons. But if you think about squeezing a water balloon, right? What happens? All the fluid moves forward. If I'm an organism, if I can squeeze my fluid forward, now my body just moved, right? So hydrostatic skeleton, you can compress against that fluid and use that to support muscular contraction and movement. If you don't have an external skeleton or an internal skeleton, you don't have any kind of a hard body surface. So if you're a soft worm, you don't have anything to compress against, but fluid gives you something to provide tension to compress against for movement. Okay. And then lastly, we classify animals using the terms protostomes and deuterostomes. And these only apply to things that have a mesoderm layer. So they only apply to triploblastic organisms. And it has to do with how the mesoderm develops. So in protostome development, a solid mass of mesoderm splits and then it grows in opposite directions and ultimately joins up. And that becomes the mesoderm layer. In deuterostomes, the mesoderm actually pinches off of the endoderm digestive cavity to form the mesoderm layer. I'm not really that worried about this. It's two different ways to get mesoderm, but it gives us some of our evolutionary boundaries in our tree of life. Okay, those things that have protostome development have the first opening in the body is the mouth. So that original blastopore becomes the mouth. In deuterostome development, that blastopore becomes the anus. And so deuterostome means mouth second. Not all organisms have an anus. And so those that develop it tend to be those that have a complete digestive tract, as opposed to those that have an in and an out as the same opening. And that brings us to our 3 phylogenetic trees that I have pasted into your notes. I don't expect you to memorize these or go through these. We're going to talk about things in an evolutionary pathway, like from our simplest organisms to our most complex organisms, and we'll think about what their relationships are. But I have given you these so you can kind of look at the groups of animals. We can't possibly go through all of them, but some of them are here. And This is a less complete layer where we look at no tissues versus tissues, radial symmetry versus bilateral symmetry, cephalization in 3 tissue layers, deuterostome development versus protostome development, cuticle that molts or not, or no cuticle, in terms of our development of our phylogenies. Okay, so these are just for you to look at and refer if you're curious how are organisms related to each other. I gave you 3 different ways to look at those questions.